Five.
Hello, everyone.
All right,
Thank
everybody,
you.
we're going to get started on the penultimate session very soon. So if we can all take a seat, maybe just a few more minutes, and then we can get started. Dr. Israni,
Got it.
can you hear us okay?
I can hear you fine. Thank you. Can you
Yep.
hear me now?
Oh, I think you're still on mute. Can we check any sound?
I am not on mute.
Oh, perfect. Now we got you.
You got me.
We got
Okay, I'm going to try.
All right.
I'm going to try to go full screen mode again.
Awesome. Thank you. Okay, so we're going to get started on the second to last session of the day, and it's my pleasure to introduce Sharat Israni. He's the NIH scholar, data scholar at UCSF and the CTO of the Bakar Institute, and has really been a leader in data science, medicine, public health, trying to make data science accessible to your average citizen. And he's going to be talking about his work on the NIH NCAT's Biomedical Data Translator. So thank you very much for joining us. Take it away.
A quick check. Can you hear me?
We can hear you. Yeah.
Okay, thank you. I want to thank the Healthcare Life Science Symposium organizers for allowing me to be here. I also regret that I couldn't be there physically. Well, I'm a long term technologist, but I've spent 10 years now in the world of medicine. It's been outstanding. And even as the possibilities continue to explode, as you can all watch them unfold, it's been a really rewarding thing. I want to, at the outset, thank Tyler Beck, who is the NCAT's program director, Biomedical Translator. He has been a great help for me to get here, but also he's been phenomenal for the translator itself. Tyler, if you're anywhere in the room, I hope there's a microphone near you because you're welcome to jump in anytime and keep me honest or answer questions right there. Also, I will disclose that I am, as was said already, I'm actually with UCSF, but here I put on, and also with UC Davis, but I put on my NIH hat for my presentation here. I was once part of one of the knowledge network, Spoke Knowledge Network, the UCSF one, which is a part of this translator. About 5 or 6 years ago, actually, we had organized a workshop at the behest of NSF and NIH in Bethesda. It was about open knowledge networks, what's the good of them, and so on and so forth. I'm going back to the origins a little bit of this story, and biomedicine was one of the key disciplines featured over there. There were four. Next thing you know, a year later, out of the president's office came this bulletin or this report from the interagency working group saying that this is important stuff. Now, going a step forward, what happened exactly 2 years ago was that a report came out from the National Security Commission saying artificial intelligence is a national priority now. I think it was presented to the Congress and things started to happen. Over there, they said there are 5 areas where it's really key to have AI be a priority. As you can maybe see on the top, it says healthcare. And if you look towards the left, you'll see science and look closely at it. That includes life sciences, including, in this case, expediting drug and vaccine discovery, as an example. Then, if you take a step further, you look at down the 750-page report, you'll find that open knowledge networks are a very fundamental component of AI in those five national areas of national importance to apply. And here we are, meanwhile, Biomedical Translator was on its way doing good things. What it does is essentially surfaces a whole bunch of data that's lying highly scattered everywhere to, in sort of a familiar format to you, the knowledge graph format. And over here, you're seeing essentially just a hairball of a bunch of green drugs and a few diseases, which are those red triangles. They sort of look like that if you really looked at it visually. What the translator does is it actually you can traverse any path. Over here we happen to have picked two source and object, and you've got tramadol as a source and you've got an object which is a particular form of pain. You can find relationships between them if you visually look at them, but of course visually is never good enough. you've got to compute a whole bunch, as we'll be showing in a bit. And this just happens to show some so-called one-hop connections. Of course, there are many intermediate type nodes that you do end up finding the need for in these drug discovery types of queries. And you'll see them unfold as we go along. Now, the problem with human biology data, or the human health and human healthcare and life science data, especially life sciences, it is really, really huge and all over the place. These are just some of the sources that were brought into the biomedical translator. As an example, you can see, I mean, if you look hard enough here and there, there's drug bank right on top, which has all sorts of drug interactions and chemical structures and targets and mechanisms that it sort of elaborates on. You've got CTD to its right, which is the clinical trials database. You, just as it sounds, down there somewhere you'll see ClinVar at the bottom left, and that is the relationship between variants in your gene and clinical outcomes or clinical phenotypes. The big one is on the top left, which is PubMed, essentially the entire corpus of um, publications related in healthcare and life sciences, which has been sort of scarfed up by various forms of NLP. These are only some of the sources, and this has been a big problem. Just drug bank alone, or any, not drug bank necessarily, any of these could be of the order of 10 to the 8 or 10 to the 9, key value pairs or relationships that you can find. It's enormous, and they're not obviously reconciled against each other in terms of sort of the link points and so on. And so just getting to a working set of knowledge graphs out of all these, resolving the differences between them, has been a major step that translator has performed over the last couple 3 years. Now, the architecture looks a little bit like this. You've got public databases at the bottom, and you've got those at least 100 that are being scarfed up right now, on which you've got knowledge providers. Knowledge providers, their job, is to go to those various public databases and sort of bring them together in a way that makes more sense. As a knowledge provider, you have one or the other discipline. It could be genomics, it could be clinical, molecular, all of the, and many more. But yours is to find where these different public databases are and reconcile them against each other, and make them clean. On top of that, there's a set of reasoning agents, each of which are driven by key questions, which we'll see emerge in a minute. And in the near term, these key questions are essentially drug discovery oriented. Each reasoning agent follows its own logic or its own mechanisms and intelligence, whatever it wants to do. All of them are brought together on top by a relay agent, basically a relay system, whose job is to essentially serve as a relay, but act, if you wish, as a federator or sort of a scatter gather, however you want to look at it, but essentially manage the queries. And in fact, I already spoke to some of this, but here you see what the genomic, three different types of knowledge providers. You can see them represented over there. At the relay system level, it also serves a UI that's emerging now, which is making it far easier to essentially query this translator. Now, here's a sense of size. We don't actually sit there counting nodes and relationships and edges, but we get a sense of what's going on over there. We estimate there are at least 100 million nodes of 62 different types. You'll see the types in a moment. And at least 300 million relationships of about 77 types. There are 14 funded teams, 150 in the consortium, funded individuals, and then there's a small intramural and developer and management team. The developer team for the intramural one has created the automated the relay system, the structure that I showed right before this. That's about the size of it. Although if I was wrong by a factor of 50%, I wouldn't be surprised. Now, describing biological relationships is really hard. It makes for very challenging syntax. These are the entities. You've got chemical entities, biological entities, and the subtrees therein. And then you've got a whole bunch of other supporting sorts of entities, administrative attributes, and so on. You also, when you try to explain the syntax, you have all these predicates, and these are some of the predicates that are in front of you. A big one is, right on top is effects. Effects is not at the leaf node level. Effects is in the middle somewhere. There's a subtree below it. There is associated with. That's a big one. Sort of. Maybe you can't see my cursor, but sort of a quarter of the way down the top, which has got quite a large subtree below it. You got causes, treats. Associated with is a very hardworking predicate, by the way. And then these predicates it looks like a little bit like this when you start to try to explain in English the subject and everything about the subject. The object, which in this case is CDKN2A, that's a particular gene, and its form or variant, mutant form, object aspect methylation, etc. And then the predicate that's involved, in this case effects, gets to be pretty elaborate. And here's just a representative of when you're trying to, this is just the enum of the sort of chemical entity or gene product aspect. As you can see, the activities could be folding, mutation rate, splicing, mutating, I guess, splicing, stability, all that. Molecular modifications start with the A's at acylation, acylation, alkylation, and so on. And it goes on for about 17 different molecular modifications. So by the time you really try to explain what's going on biologically, it gets to be mind boggling. And yet you have to do that in order to essentially compute. Trying to advance. Now the user interface is under construction, and over here you see the 5 initial canonical queries that are given. What drugs may treat a disease, or what chemical / drug upregulates or downregulates typically a gene protein, or vice versa. And then when you do that, it gives you a little bit of autocomplete help. In this case, Duchenne muscular dystrophy. It gives you all these other alternatives in the sort of named entity called disease, and you can pick one and go for it. What chemical downregulates the FLT3 gene mutation, and as you can see, there's not just human but other organisms as well, all of which is important to a biomedical knowledge network for research. Now, the problem is that with big data come big results. And I want to dwell a moment on this slide and tell you exactly this and the next 1 or 2 slides, and then I'm kind of done. A typical researcher way of thinking to discover a drug is to say what chemical or drug, in yellow, will treat rheumatoid arthritis. That's the one on the right in red. But they know what the pathway that really is, they think pathway first towards the end. What do they know about rheumatoid arthritis? They know, for example, MyD88 is sort of an inflammation causing pathway, biological process. So they say, look, tell me what chemicals or drugs up or down regulate genes that have an effect on the pathways through the gene-protein interaction, protein-protein interactions and pathways onto rheumatoid arthritis. And then now that I know that much, that pathway is very interesting. Are there any approved drugs that deal with that pathway also? In this case, imicumod, which is used for a particular form of skin cancer, actually, keratosis. And so if you look at that, what happens, you do a query for chemical drug for rheumatoid arthritis, you may find there are 20 pathways, 30 genes per pathway, 30 chemical entities per gene, or you may have 10 approved drugs. This results in easily 18,000 combinations to evaluate. And I could be wrong by a factor of 4 over easily. You could easily end up with 500,000 even combinations by the time you go through the knowledge network and all the data it's given you. So the problem becomes, how do you actually order the results and make them more relevant, sort of in sort of like search engines do? And that's just to evaluate them. Then you evaluate those each of those results. Well, what we do is we consulted with some research experts, subject matter experts, and we said, what are the things that cause you to think about how to look at a particular possibility, a drug target? And over here, you say, you see that what the question again is, what drug may treat disease X? And the challenge is that you've got all these diverse variables of various levels of utility, I call them, which you'll see in a moment what they are. And you're trying to make them independent of each other and yet come up with sort of a function that gives you a sort of a result score for a particular result. Say imicomod treats that, uh, treats whatever, treats even rheumatoid arthritis. Okay, now what we do is we take the different reasoning agents, and remember they're semi black boxes to us. We take the different reasoning agents and come up with a function to resolve them firstly. And then we take other things that have so far been declared as interesting by the SMEs, which include clinical evidence. Do you have any clinical evidence for this? Drug stage, which really should be clinical trial stage, but for drugs. Nevertheless, that's what we mean. And then an all important novelty factor. Now, there's some function that combines all these into a result score. And I'm going to sort of jump to the end because, of course, we don't have a lot of time. Um, these are on the right. You can see the PIs who are leading the thinking around this result scoring. The G factor, once again, the G factor was the one which was used by the six different reasoning agents. Is used. They use some dimensionality reduction to inform. What are the weights we should give these different reasoning agents depending on the query type. And one of the issues we run into pretty soon is that the results are pretty sparse. But we're finding mechanisms to increase the result density by sort of essentially getting similar edges, similar results to be the same for the purposes of scoring. And that's one thing. One thing we're doing in various forms of dimension reduction. Then looking at the rest of the function, once again, the rest, actually, I don't need to go back. The rest of the function right on top, which combines the agent scores G function with the other three factors. We are looking at them pretty carefully. Looking right at the bottom, you'll see we're considering some fuzzy logic approaches. And for those that are familiar, dysgenet, which is disease gene network, has some utility based scoring based on what SMEs like to see. I argue that the fuzzy logic we're using resembles that. As an example, clinical evidence is the factor called clinical evidence is a weighted odds ratio of association strengths between entities with drug and disease. If there's clinical evidence, how strong is the evidence? How strong is the association? And then drug stage or clinical test trial stage is discrete, 1 through 4, as we kind of all know. Novelty factor has got a whole bunch of very interesting components. There's the publication count, the publication recency, a sort of a cross product between the two, inferred relationships other than just the direct relations stated by, say, drug bank or some molecular source. Is the drug being used off label? Is there any lack of clinical evidence? And we're sort of teasing through all these things to see how, what importance the subject matter experts give these different components. And by the way, what are the interplays between them, which I could describe, except I want to save some time for comments and questions. And so when you go forward, we look at, for example, this is what a result score may look like. It may look like the sort of eigenfactors that came out of the G function resulted in two agents getting, these weightages 0.86 and 0.57. But the other agents might not be very important because they all follow different logic to this drug treats disease type query. And let's just say based on what the result came back with, the agent scores are 42 and 70 and so on and so forth. The clinical association strength, like I said, is the association strength. It may be 30 based on some SME utility scale, and putting that number in, you may end up saying, okay, this result, that drug which I can barely pronounce, treats AML with a score of 99.82. And we've got, as I mentioned, for hundreds or thousands, maybe even tens of thousands of such results per query. And in the end, it starts, and this is a very, very, very early version of the UI. Then the end, this is what it looks like. It gives you a direct link. This our drug treats AML, but also you'll soon be seeing, if you go below this, actually a bunch of multi-hop paths as well. And then on the left, you've got this faceted search where you can start to. This is what the experts like. They like to group things up, group things down. They like to eliminate, known drugs, etc. Okay, last couple of slides. What are some key system challenges? As I've been implying all along, there's little use to giving a researcher 10,000 results. What are they going to do with them? And so, what are the strategies for us to intelligently surface the top 20, shall we say? You of course can think this. You can be forgiven for thinking this looks a little bit like web search, and it does. But the difference is the researchers have a whole different mindset on the accuracy and explainability of what you're showing. Anyway, so that's a challenge. Performance, when you've got a system like that created by 14 different entities around the country, you can imagine the whole thing is never optimally architected, and certainly not for performance at the first go, although there's a long road ahead, and I think the performance is getting better and better already. But that's an issue to always watch for. And then as we take on other questions, like other questions than what drug treats disease, you including gene regulation, then you have to adapt the F function as well. For example, genes have 4 different levels of novelty in their target, and we've got to bring that in, T dark, T clin, and so on. I want to end with an inspiring story about David Feigenbaum. Um, David Feigenbaum was a medical student when he found out. In effect, it didn't happen in one quick clinical visit. Let me tell you, he found out that he had what's known as an idiopathic multicentric Castleman's disease. And Castleman's disease is the short form for it, and it's a very rare disease. Of course, it was killing him. It was killing him, and there was no drug for it. But what he did, he decided, look, I am qualified enough to do my own research. And he found through his research using mechanisms like the translator, and he's been to the translator and he's been an inspiration for the translator. um, he found out that his mTOR signaling was being sort of down regulated. That wasn't a good thing. So he was on a quest to discover an mTOR inhibitor. Now, mTOR inhibitor, when you have such a rare disease and you have so much data, you can never assume that somebody has studied the condition carefully enough to be able to come to a solution. But he did. To cut the long story short, today he's a faculty member in Penn Medicine, and he wrote this book called Chasing My Cure. By his own account, he has lived about 112 months past when he was supposed to, which is nearly 10 years. You think about it. It goes to show how one individual can make such a huge difference, and our goal with the translator is to enable many such individuals. Thank you. Any questions, comments, feedback? We welcome it.
So I have Tyler sitting right next to me, so I'm going to give you the first opportunity to see any follow-on.
Sure. I mean, Sharat did a great job of explaining the system. I'm Tyler Beck. I am the program director for the Biomedical Data Translator at NCATS. I think the only other thing I would say is that we've been able to build this system and do plan to have a public release of it by the end of this year. We've built the system using less than $50 million worth of funding. It has been NCATS has a small budget comparatively to other NIH institutes, and we've used that budget to the best of our ability in this program. And we've created a really interesting and very inspiring community of researchers on this program, which I'm happy to talk more about to any of you. But I want to give Sharat the full question and answer here. So thank you.
Thank you, Tyler.
Tyler, by all means, jump into the Q&A as well, and thank you for surfacing. There's a question in the chat. How does the novelty factor not fall into the trap of irrelevance? Because, as I understand, it considers the lack of evidence as a measurement of novelty, but it is not possible to be completely irrelevant, uncertain relations. Slightly parsing that how I can, the lack of evidence is not the only measure of novelty. It means that if something is being indicated as it could work, but is not given, then by definition, that's a one signal of novelty. Besides, there are others. It is not possible to be completely irrelevant / uncertain relation. Yes,
Sharat, can
yeah.
I add to that just a little bit? Sorry, this is Tyler. So the novelty factor comes into play really importantly when we talk about rare disease, because NCATS does focus pretty heavily on rare diseases, and in a lot of cases, those patients who have a very rare disorder have been treated with everything under the sun that their physicians can think of. And what that means is that if you're looking to find new treatments for a rare disease, you want to skip past all the things that have been tried already. And so that's sort of where that novelty factor comes into play. We imagine that we expect that some of our researchers will want to say, show me only novel results, not things that have been approved for similar diseases, things like that. So that's where that comes in, and it will be important for the overall score, but will be probably more important for this sort of faceted search aspect.
Perfect.
So I see there's one more online question. Do we want to do that one first, and then we'll hand it over to Deb?
Yes, sure. Are there applications of this system to reduce mental health disparities? The system could very well be ready now for someone to go query it for. I mean, all the disease conditions are essentially in there. Has anyone done this? I don't know, actually. But it'd be worth a try to go in and see what it shows.
Great talk. This is Deborah McInnes. I wonder, and that story about Feigenbaum was like really inspirational, but I wonder what it takes for somebody who's not quite as well trained, like isn't a doctor, to be able to use this to help either themselves or their loved one, potentially in collaboration with the doctor. But what kind of steps does it take for broader usage of this?
Fantastic question. One that dogs us every day in our lives as sort of systems creators. I'm going to let Tyler end what I say with the introduction that actually even the subject matter experts said, I don't want to investigate this by myself. I want someone next to me who can help me navigate the system. And I don't think they'll be able to ask the questions I'm asking. After all, I'm the researcher. So they see this sort of as a tuple or three-tuple or something like that. Right now, I'm myself helping someone who's been found with a rare disease, who is a doctor himself, to discover what could be some solutions for him. I'm able to easily navigate it, but I don't know what he's asking me. That's sort of a half answer. Tyler, anything to add to that?
Yeah, I mean, the other thing I would say is that we, as I said, we expect to do a public release of this toward the end of the year. There will be a very clear indication when we release that this is a tool that's being built for researchers, not for clinical decision support, because we don't want, a physician to go and use Biomedical Data Translator to find a potential treatment and just immediately say, okay, let's try it. We don't want that. We want this to be used in a research fashion. So clinical researchers may find it very valuable, but we don't, and we definitely don't want the general public to start using this and say, okay, I'm going to treat myself with this because NIH says to. That would be terrible. And so we will make it very clear that this is a research tool, and it will always be meant to be a research tool. Now, that doesn't mean that systems like this, or even this system eventually, could be adapted more clinically. But for now, we really want to focus this on the fact that it is meant for researchers.
Okay, Eric, you can have the last question.
Thank you. Wonderful talk. Sorry. Wonderful talk. One thing that I'd seen that worked very efficiently is that if you provide a lot of relevant, succinct information to an area like genomics and cancer, to a virtual tumor board, you have a lot of the minds there with all different backgrounds, and then they can hash through the evidence and the literature and really inform themselves and come up with good prospects of what could be tried. Have you thought about using Translator for virtual tumor boards or virtual any other kind of clinical groups?
Sharat, is it okay if I answer that one? So
Please.
this is actually happening every day at the Precision Medicine Institute at the University of Alabama, Birmingham. They see a lot of rare disease patients and patients who have been treated with, again, everything that could be tried. And they're using, they're using both Translator and LLMs now in concert to try to find new potential treatments for patients there at the Precision Medicine Institute. Dr. Matt Might is our sort of star there. He's been using Translator for since we started the early phases of it, and they have some really amazing stories of treating patients with seemingly innocuous, I wouldn't even call them drugs, just different things that are household agents. We have one case where a young woman was had cyclic vomiting syndrome, and they used translator tools to try to find that if she would sniff an alcohol-soaked Q-tip, it would stop her nausea. And this young woman had been throwing up every day of her life for 19 years, and suddenly had no more nausea after that when she would just take a little baggie out and sniff some isopropyl alcohol. It was amazing. Stories like that are what we are hoping to continue building with Translator.
Thank you, Tyler.
Thank
And
you,
thank
Tyler.
you, everyone.
Thank you, Sharat. Okay. So our next speaker is Steve Hamby. Steve's an experienced teacher, software developer,